to explain you the following work entitled Mechanical or Biological Control of 𝘾𝙧𝙧𝙦𝙧𝙧𝙀, which will be more effective based on a publication from last year. Let's go! Anthropogenic movement of its species over the past centuries has favored the establishment of invasive non-native plants worldwide. In recent decades, concerns about environmental problems produced by invasive plants have aroused the interest of researchers, warning about their impacts. For example, plant invasions can induce native or endemic species turnover, disrupt nutrient cycles, modify trophic feeding guides, and soil physicochemical properties, etc., being the major driver of habitat degradation, especially in islands and in coastal mainland areas. Furthermore, invasive plants get rid of their natural enemies, conferring them a competitive advantage over natives by being able to spread quickly and easily. 𝘾𝙧𝙧𝙧𝙦 is one of the most problematic invasive species worldwide. This succulent and clonal plant, native to South Africa, is frequently found in coastal areas and dune systems, where it drastically affects the biodiversity and the functioning of these ecosystems. Consequently, establishing management strategies should be a priority to slow down their rapid growth and spread. Nowadays, diverse management strategies that focus on mechanical and chemical control have been implemented. However, they are expensive and requires much labor, and in turn, they can affect the environment and other native species. In the short term, this combination is not entirely effective, since the fragments of 𝘾𝙧𝙧𝙧 can be rerouted after hand removal extraction. Taking all of this into account, our objectives were 1. To assess whether gram surface type influences the senescence process of the plant after uprooting, and 2. To explore the effectiveness of specialized herbivores as biological control agents. In my case, I used 𝑘 mesembryanθ scale in 3 different scenarios. We placed 60 clonal fragments of 𝘾𝙧𝙧𝙧𝙧 individuals into pots, and they were allowed to be rooted for at least 16 days. After this time, we uprooted 40 individuals, which 20 were laid on sand and other 20 on stones, to avoid contact with the sand. The remaining 20 were left rooted. These are my 3 scenarios: rooted, on sand, and on stones. Then, we inoculated 30 individuals of 𝘾𝙧𝙧𝙧𝙧, 10 of each treatment, with 1 female pulvinariella per plant, while those of other 30 were not. For about 13 months, we measured how soil surface and herbivory affected plant physiological activity by measuring several measures of fluorescence, Phi2, photosystem 2 quantum efficiency, Phi-NO, measure of unregulated energy dissipation as fluorescence, and Phi-MPQ, measure of regulated energy dissipation as heat. And also, we determine plant survival and rerouting by using a visual rating scale. After 13 months, we harvested all individuals of 𝘾𝙧𝙧𝙧 carefully, and they were taken to the laboratory. We counted the final number of pulvinariella, and we determined several biometric parameters on fresh and then on dried plants. After this, we crushed the dried shoot and root parts, and we determined the shoot and root nutrient composition to find out different aspects of the plant, such as the degree of decomposition. Finally, we determined total phenolic and tannin contents using the Folin and Sicolto method. And what did we find from those analyses? As an aside, dashed lines indicate that the plant was under the influence of pulvinariella, while solid lines do not. Well, for the photosynthetic parameters Phi2 and Phi and O, a decrease was seen in all cases except for rooted control plants. This indicates that harmful byproducts for the plant are being produced over time and probably inhibit photosynthesis. For Phi and PQ, an opposite upward trend was observed, except for rooted control plants. This suggests that plants attacked by the insect were defending themselves from the stress they are undergoing. Moreover, this is related to the amount of magnesium in the plants. Our results showed that Carpobrotus individuals under herbivore attack had a reduction in it, which may explain a decrease in photosynthesis since it is an essential ion in this process. Survival of Carpobrotus under the pressure of Pulvinariella was reduced by 100% when they were on stones or sand, and by 90% when they were rooted. Furthermore, survival of individuals without herbivory was reduced by 100% when they were on stones, and by 70% on sand. The most pronounced decline and rise in photosynthetic parameters occurred between March and June, especially for individuals under the attack of Pulvinariella. Furthermore, this could be seen in the survival graph. Our results show a severe reduction of survival from the 8th month, April. In contrast, for those on sand and stones without the herbivore, plant survival was reduced from the 10th month, June. This is related to the life cycle of Pulvinariella, which has two population peaks, one in spring and one in late summer. The decrease in plant growth due to the difficulty of replenishing the resources eliminated by the insect and increase in stress also affected the herbivore itself, causing a decrease in its number according to the capacity of the plants to recover the nutrients consumed by the insect. For routine monitoring, our results indicated a significant difference depending on the ground surface type. Rooted individuals, as their designation indicates, remained rooted until the end of the experiment. Individuals on stones did not get rooted, and some of those on sand did, of which those under Pulvinariella's attack showed greater rerooting. The attack of the specialist herbivore severely decreased the performance of Carpobrotus, resulting in lower growth rates. However, our results did not indicate an appearance reduction in root growth when Carpobrotus was on sand, which could be explained by the increase of sodium in roots. One of its functions is the growth stimulation. Carpobrotus individuals on sand and stones had a more significant amount of nitrogen and phosphorus than rooted ones. This could be explained by the fact that plant decomposition is usually positively correlated to the increase in them. In addition, Pulvinariella as biological control could advance, in almost 2 months, the decomposition process of the plant. The attack of Pulvinariella may trigger an increase in carbon and hydrogen, which can be related to carbohydrate biosynthesis like total phenols and tannins, which are chemical defenses, compared to unattacked plants. So, our study highlights that placing Carpobrotus edulis over an inner ground surface after its uprooting favors its death, avoiding the problem of rerooting after plant hen removal. And the specialized herbivore Pulvinariella caused the death of Carpobrotus in all studied cases, regardless of the ground surface type where the plant was placed, accelerating its death in almost 2 months. Therefore, we conclude that the best ways to manage Carpobrotus are direct inoculation with Pulvinariella, or even the combination of placing the plant over an inner ground surface while it is under attack by herbivores. This could help land managers and authorities to implement appropriate management strategies to avoid rerooting after mechanical control. Thank you.